Good afternoon, everyone. It's my great honor today to present our Grand Brown speaker. It's my mentor of approximately 25 years. He's been mentor and grand mentor to many people over here at Yale, Dr. Mario Sznol. Born in Cuba, raised in Texas, he got his undergrad education at Rice University, his MD at Baylor, internal medicine at Mount Sinai, and he did his fellowship at the National Cancer Institute. In 1999, after his fellowship, when he was approximately 12 years old, he joined the investigational drug branch and CTEP at the National Cancer Institute, and he did his clinical work in the surgery branch under Dr. Steven Rosenberg. I think that's where he was introduced to high-dose interleukin-2. Between 1994 and 1999, he led the biologics evaluation program, where he assessed many first-in-human drugs, such as some that you're all very familiar with, trastuzumab. In 1999, he came to New Haven, where he led a biotech company called Vion. He volunteered in the melanoma clinic for a half a day a week, and that's where I met him. I was a fellow. Essentially, Dr. Sznol needed a permanent fellow. A temporary one wasn't good enough. So in 2001, he convinced me to become his permanent fellow, and the rest is history. That was a decision I've never regretted. Today, he's going to talk to us about his vision for immuno-oncology, both in our group and at Yale in general. I'd, however, like to take just 1 minute to talk about his past. So in 2001 or 2002, the median overall survival for metastatic melanoma was in the order of approximately 9 months. There were a few exceptions, such as the high-dose interleukin-2 treated patients, and that was a program that Dr. Sznol had set up over here at Yale. But in around 2008, the median survival was starting to improve, mostly because of the advent of anti-CTLA-4, both in clinical trials and in expanded access programs. In 2008, Dr. Sznol started the first in-human study of what is now known as nivolumab. This was a multi-center trial, and Dr. Sznol was the site PI. Some dramatic and prolonged responses were seen, not just in melanoma, but in other diseases as well. So Dr. Sznol then took the initiative to convince a company called Medarex, which owned both of these drugs, to allow him to combine the two. And this was the birth of the Ipi-Nivo combination regimen with all of its challenges. So now, in 2025, Ipi-Nivo has cured thousands of patients with melanoma worldwide, and it's also cured a good number and prolonged survival in patients with other tumor types as well. However, for Dr. Sznol, this is not good enough. We're going to cure 100% of the patients with 100% of the diseases, and he is going to tell us precisely how to do that. So without further ado, thank you so much.
Thanks, Harriet. That was a little embellished, but nevertheless, thanks anyway. Just want to start first. Just want to say a couple of words about Dr. Edward Chu. Ed died just this past week. And before I start, Ed played a major role in many of our careers. He's a giant in the field. Ed came to Yale in 1996. He was at the VA and became chief of medical oncology in around 2004. He was here through around 2010, then went to be chief of medical oncology at Pittsburgh, and ultimately became the cancer center director at Einstein. Ed was a great friend, and one of the most magnificent individuals that I've ever interacted with. I remember when he was chief of medical oncology, he was one of the strongest supporters of everybody who was in his group. It didn't matter whether people supported him or not. When they came up for promotion, Ed was the one who went to bat for them 100%. He was really a great person. Smartest guy. I've never seen anybody write grants like Ed wrote grants. I mean, he was just amazing. It's a huge loss for our field and for the world in general. Lori's on the line, his wife, and I just want to send our condolences to Lori and to his kids, Josh and Ashley. It's an enormous loss. By the way, Ed was here when medical oncology wasn't what it is today, and he helped transform it and sort of paved the path for what Roy Herbst ultimately came and accomplished here at Yale. Let me just provide my disclosures, and I'll start with the talk. Some of the things that I'm going to talk about, I have a financial interest in. Probably doesn't matter anymore, but these are companies that I'm interested in. I'm going to talk a little bit about some of their agents during this talk. So at the beginning of the talk, I'm going to talk a little bit about science. At the end, I'm going to show some pictures of our group, a little personal thing, but I'll try and get through it all, within the 60 minutes here. And I'm going to fly through some of these slides and data. So first of all, anti-PD-1 has really changed the world. It provides clinical benefit across really a broad range of solid tumors. There's almost no solid tumors where we don't use an anti-PD-1 or anti-PD-L1 agent anymore. We use it in advanced disease and after post-chemo radiation, in the adjuvant setting, the neoadjuvant setting, almost in every imaginable way in the clinic. We've tried multiple combinations in the clinic, but there are now only approvals for approximately 3 or 4 agents. It's been approved in combination with anti-CTLA-4, with anti-LAG-3, which is relatlimab, with VEGF receptor inhibitors, with chemotherapy, with antibody-drug conjugates, and with some targeted therapies. But really, we haven't really made major advances with these combinations. They add something, but not a lot to anti-PD-1. One of the major limitations in the field is that we don't have predictive biomarkers for when this is going to work. We know some things. We know that if the tumor or the immune cells in the tumor express PD-L1, you have a better chance of responding. We know that if you have a high tumor mutation burden or mismatch repair deficient tumors, that you have a higher chance of responding. And we know in sort of lab-based assays that if you have an interferon gamma gene signature, you're more likely to respond. But what's missing is that we don't know in any individual what that next agent is that you need to add in order to get better activity. And we still don't have that today. One of the reasons is that there's many mechanisms of response. It's hard to identify the mechanism of response for any individual patient, and the biology is incredibly complex. There are multiple leverage points here, and knowing which one is important, which one is going to give you to that threshold effect that will cause a tumor to regress is still very hard to define in any individual. So in order to move forward, we need to understand a little bit, not just what the mechanisms of resistance are, but how these drugs work. Some of the work has already been done, but even that really hasn't identified a clear path forward. Some of the things that I'll talk about today perhaps won't involve combinations with anti-PD-1 at all. These are the key drivers of immune therapy malignancy. I'm not going to go through this in detail. We know that something has happened in the tumor, some sort of an innate immune system needs to be activated, like STING. It's either in the tumor cells or in the antigen-presenting cells. Somehow that leads to interferons being produced and chemokines within the tumor microenvironment. And at some point, the tumor cells die, tumor antigens are taken up by dendritic cells that are in the tumor microenvironment, and they're either transferred to a draining lymph node or to tertiary lymph node structures within the tumor. And then there's an interaction between an antigen-presenting cell and a T cell. And probably most of the agents that we give today, that whatever we're manipulating is probably occurring at the APC T cell interface. But there's another place where things happen, which is when those T cells actually get to the tumor. And then there are tumor T cell interactions. And for that, we really understand a great deal less. We know that those T cells can kill. We know that they can make interferon gamma. We know that they make chemokines. We know that they activate other non-immune T cells, non-T cells, immune non-T cells in the tumor microenvironment. And somehow that leads to tumor regression, and whether that's vascular collapse or blockade in something else. We just don't know what the process is beyond that. But one of the things I think we're missing is really a clear understanding of what happens at the tumor T cell interface. For many years, we thought that anti-PD-1 was really working at the tumor T cell interface. More and more data over time suggests that it's really at an APC T cell interface where anti-PD-1 has its major effect. So why is it that patients don't respond? And theoretically, there's just, and I've given this slide talked about this slide 1,000 times, but theoretically, there's just really two big buckets. Those are the patients that don't have any T cells in the tumor microenvironment. So it's either an immune desert or the T cells are excluded, or the other is the T cells are there. There's an antigen-specific T cell response, but the tumors still don't respond to immune checkpoint blockade. And then you can go down to multiple deeper levels. Why is it that there are no T cells there? And its most likely explanation is that there's no priming either. Nothing has happened. You haven't really had an APC T cell interaction. You haven't developed T cell responses, or those T cell responses are diminished, gone away. Or the other possibility is that those T cells have been generated, but they just can't get back into the tumor. And then you can look at even deeper levels than that and ask, why does that happen? And it could be anything from there are no antigens there, or you're homogeneous at HLA alleles, or the antigen presenting cells have been suppressed or inhibited or not activated. Or maybe there's the chemokines that you need to get the T cells there aren't there, but there are many different reasons, and there's probably even another level beyond that that explains why those things happen. But again, trying to understand what that is in any individual patient is very hard. Now, if the T cells are there and you don't have a response, you can also ask, why is it that they're not responding? And there's many potential reasons. One is maybe a numbers game. There may not be enough T cells there. The other is that they may not even be tumor antigen specific. The other is these might be very low affinity T cells. They can't recognize the tumor. It could be that the T cells get in the tumor and don't differentiate into effector cells that can kill or produce cytokines. And then there are many other issues. You may not get the T cells from the lymph node back into the tumor. It could be that the microenvironment is very hostile. There's not enough oxygen or glucose for those cells. It could be that the tumor or other immune cells are making inhibitory cytokines that prevent the cells from working. And we already know that things like prostaglandins and adenosine can inhibit T cell activity. There can be other ligand receptor interactions besides PD-1, PD-L1 that can block T cell function. There are multiple cells that are within the microenvironment that can inhibit those T cells. And finally, it could be that the tumors just simply downregulate the ability to be recognized. They can lose beta-2 microglobulin, they can lose adhesion molecules, or they can lose or downregulate class 1. So the number of mechanisms, theoretical mechanisms, which can be the cause for a non-response to immune activation modulation is enormous. And again, the major limitation in moving forward in the clinic is it's hard to identify in any individual patient which of these is the most important, and whether if you modulate that pathway, whether that's a critical and non-redundant pathway that will lead to tumor regression. So when we think about improving anti-tumor immunity, we can put it into sort of two buckets. If you have a tumor that either can't be recognized or it's non-immunogenic, then you have to look for synthetic ways of going about doing this. You can either use CAR T cells or you can use CD3 engagers, and we'll talk a little bit about CD3 engagers. The other thing that you can do is you can try and take advantage of innate immunity like monocytes, macrophages, and NK cells and combine them with standard reagents. If in fact the tumor has generated a T cell response that's immunogenic, then there's a number of ways that we can do it. If it's a numbers game, you can try and expand that T cell response. If it's because there are other inhibitory pathways, if we knew what those were, you can block those other negative regulators of function. If it's a problem with T cell migration to the tumor, there's now many different molecular pathways that can block T cells from getting in the tumor. There's a company that was actually developed by Liping Cheng and multiple molecules that we know can do this. And the final thing is you can just enhance the tumor cell sensitivity to immune effector killing, but we don't really know how to do that, and there aren't many approaches for that in the clinic. So where can we focus in the clinic? And my main interest is really in trying to get drugs into our clinical program that will help patients, is where can we intervene right at the moment? And one of the things that we can do in the clinic is give cytokines. Cytokines are very important molecules. I'm going to show you little data that can really impact the function of T cells. And it's possible that cytokines might address some of those mechanisms of resistance, including the numbers game, there aren't enough T cells, or maybe even situations where the T cells don't differentiate into effector cells, because cytokines can also cause T cells to develop effector function, which might make them more effective in the tumor microenvironment. The other, and there's a relatively, I think, important and interesting development over the last few years, have been these T cell engagers, which are basically bispecific molecules that can bind to the tumor and also to T cells. And by that interaction can activate those T cells, nonspecific T cells, to kill the tumor cells. That doesn't require an adaptive immune response. You don't have to have a TCR, peptide MHC complex recognition. All you need is a viable T cell and a molecule that can sort of redirect that T cell killing to the tumor, and you can get activity. Now, this has been there's been plenty of proof of concept for this in hematologic malignancies, but in solid tumors, this is becoming a more important concept. So we now have a couple of agents, and I'll show you, which have shown really meaningful activity in the clinic. One of them we're studying here, and I think will make a difference in kidney cancer. And I think these T cell engagers may be the next wave of effective agents with immunotherapy. And the beauty of all this is that we may be able to combine the cytokines with the T-cell engagers and get even more effective activity. And that's really where we've been directing most of our efforts in the clinic. Now, there are other things that you can do. You can maybe address that tumor T-cell interface problem. I don't know exactly how to correct the MHC class 1 reduction problem, but things like interferon gamma, other cytokines can upregulate class 1. There's loss of adhesion molecules, and I'll show you some data with that. And there may be a way by creating molecules to restore that adhesion between the tumor cell and the T cell in order to make that interaction more effective. We may be able to find tumor antigens that are not already presented. So there may be tumor cell antigens that are not neoantigens, and they're not shared cancer testis antigens. There are other kinds of antigens that are now being discovered, which may provide antigen-specific immune responses, and we may be able to immunize against those antigens. And David Braun, who's here in our group, is thinking a little bit about those approaches. There may be ways of doing better delivery of these molecules to the tumor. So we now have these molecules that can go directly to tumor. We may be able to attach immune modulators directly to those molecules so that we can get specific activation within the tumor microenvironment. As I said, there's a T-cell migration problem. We now know there are multiple molecules. It's going to be hard to bring those agents into the clinic, but it's another, I think, important approach. We can modulate other immune cells within the microenvironment, and we're very interested in things that can affect the myeloid cell populations. And we've recruited people here to Yale that are working on those, and single agents and combinations of agents that affect those myeloid cell populations might actually have an impact in the clinic. We can develop better biomarkers, and we're working on that. And then, of course, there are cell therapies which are being developed, which will have some promise, have substantial promise. And as the genetic engineering becomes more and more advanced, that will, I think, improve outcomes in the clinic also. So let me talk a little bit about things that we are interested in the clinic. So a number of years ago, someone called me and said, you know, we have this molecule that can target IL-2 directly to T cells. Why is that important? Because IL-2 is a molecule that has a lot of toxicity. If you give it systemically, it can work, but you can never get to high enough doses to have the sort of effects it has in animal models. And it has, you know, as you know, it causes sepsis and sepsis-like symptoms. And sometimes we have to treat people in the ICU and they develop hypotension. And it's just not a pleasant thing to do to patients in the clinic, although it can be effective in a small number of patients. But the concept here is that you target the IL-2 specifically to the cell that you want. So this is a bispecific that was made by this company, which targets CD8 T cells directly, and attached to it is a modified and attenuated IL-2. So when you give it systemically, this attenuated IL-2 molecule doesn't have much effect. But when it binds to the tumor cell, that IL-2 interaction is a high avidity interaction, and you get very nice IL-2 effects specifically on CD8 T cells. This was developed by Ivana Djuritic and Andy Young. They were protein engineers at Pfizer and developed this company. And when they called me, I said, well, this is kind of an interesting idea. Show me more data. And in fact, the data, the more and more data they showed me, the more and more interesting it became. It can have the advantages of overcoming these dose limitations of other IL-2 molecules. And what it does is if it reduces toxicity, it allows us to combine these agents with other cytokines, and that's a future plan. And I'll talk a little bit later about why I think that's important. So this is, I'm not going to go through this slide in detail because I won't have a chance to get through the whole talk. But if you just take a look, does the mouse work here? If you take a look here, you can see that many of the T cells, after giving this agent in monkeys, many of the T cells are proliferating. You can see here that AB248 activates CD8s, but not Tregs. And you can see here that compared to a wild-type IL-2 or the non-alpha IL-2s, this cis-targeted IL-2 has very significant activity in mouse models. So this is a very interesting agent and deserved to be taken into the clinic. And in fact, a phase 1 trial was launched. Dr. Kluger was the PI here at our institution. None of the data actually have been reported clinically, but I can say that it does exactly what you think it would do. It's fairly well tolerated. It causes remarkable proliferation of CD8 T cells in the peripheral blood, and the few biopsies that have been done have shown substantial increase in T cell infiltrates into the tumor. And it's an active agent. So in patients with resistant refractory disease, many of the patients have been treated here at Yale by my colleagues. We've seen real activity in patients with melanoma, both cutaneous melanoma and mucosal melanoma. Now, that activity may not be enough to get to registration, but it's real proof of concept that an agent like this can work. And the key here is not to use this agent by itself, but eventually to use it together with other agents. So are there other agents like this that can have similar activity or can be combined with the 248, the IL-2 molecule? So this is work that was done not at the company, but here by Nick Joshi and Ken Kui. Ken Kui was in the lab here at Yale in Nick's lab. She actually spent a lot of time in my clinic. Then she went on to Mass General to do a residency, and now she's doing a fellowship at Memorial. She's just a brilliant young woman. And this work, which was published from Nick's lab, showed, and I'm not going to go through the whole data. It involves activation of CD4 T follicular cells when they interact with B cells, which then produce IL-21. The key is that the production of IL-21 by these CD4 cells was critical for tumor control. And it's critical for enabling the effector function of CD8 T cells. So IL-21 is a very important molecule to get those T cells from that central memory sort of differentiation phenotype to that effector phenotype that can actually kill. What IL-21 actually does is it increases perforin and granzyme, the killing molecules inside those CD8 T cells. So the company, Asher, did a very interesting experiment and to show how you can differentiate these cytokines. So they basically took MC38, and at day 14, they treated with they either gave no treatment, they gave the CD8 IL-2 molecule, or they gave the CD8 IL-21 molecule. So basically no treatment, the CD8-directed IL-2 or the CD8-directed IL-21. And then 3 days later, they harvested the tumor, and they did single-cell RNA-seq and looked at what cells were being expanded. And so this is the single-cell RNA data. You can see control. You don't get much activation of T cells in the microenvironment. This is the IL-2 molecule, and you can see that almost every one of these T cell subsets was expanded. This is the IL-2 with FTY720, which keeps the T cells from the lymph node from getting into the tumor. So most of the expansion here was really related to T cells that were being expanded in the tumor microenvironment. What I want to point out here is that the IL-21 molecule really expands a very different subset of T cells, right? So IL-2 expands all of these T cells, but not this group down here, which are really more of the effector-like T cells. IL-21 actually does expand this group, and much differently than does IL-2. So the question is it different in mouse tumor models? And for me, the big question would be, is there a possibility in the future that we can combine these two agents together? So the other experiment that they did that I thought was very interesting was looking at the clonally expanded population within these tumors. And the clonally expanded population would be the ones that are expected to have the tumor antigen-specific T cells. And what you can see here is that IL-2 did expand these sort of colored dots, or the clonally expanded populations, which are in controlled, mostly here in group 7, which are exhausted CD8s. When you give the IL-2, you see you get expansion in a number of different subsets of T cells. When you give FTY720, not much happens. But again, you can see that the IL-21 expands a completely different group of T cells. And these are the T cells. These clonally expanded T cells are probably antigen-specific T cells. Gosh, I'm so sorry. And also the ones that are now have effector-like function. So they went on and looked at the anti-tumor activity of the cis-targeted IL-21 in mouse models. It was active across quite a number of mouse models. And what I'm not showing here, but I can tell you, is that the pattern of anti-tumor activity in the mouse models was very different between IL-2 and IL-21. So they're very different molecules that have different anti-tumor activity. So we became very interested in this, not just because we wanted to study this as a single agent, but because we thought that we could have ultimately combined it with anti-PD-1 and combine it with the IL-2 molecule, which is where we're headed in the future. And in fact, the company at this point did not have a lot of money, did not have the resources to start this trial. And so I approached them and said, well, maybe we can do it at Yale. And I said, Dr. Kluger, please help me out here. And um, like she always does, Dr. Kluger found a way to get the study written and activated, negotiated with a company to bring it to Yale. And I want to give a special thanks to Dr. Ian Kropp because it takes a lot of foresight to be able to take on a study that is investigator initiated for which you don't get a lot of money. So we're going to lose a lot of money on this study, but we saw the future of this and we may be able to make a real impact. And we're the only institution in the world that actually has this study open. And I don't want to take away Dr. Kluger's, next, the big reveal at the next talk, but so already at the second dose level, Dr. Kluger treated a patient, I didn't show the slides, but the second patient we treated with a Merkel cell carcinoma who had progressed on anti-PD-1, may not have been completely anti-PD-1 resistant, but nevertheless had progressed on anti-PD-1, had developed bad myocarditis, myositis, well, at least bad myositis, maybe myocarditis, with a single dose of anti-PD-1, and who came to clinic with a very large retroperitoneal mass that was FDG avid. At the first tumor evaluation, the mass is gone, FDG activity is gone. Actually, it's a complete responder as far as I can tell. So amazing activity already in the second patient at the second dose level. So we're very excited about this compound, not just using it as a single agent, but in multiple different ways. So the way I see things moving, at least in our unit, is to complete the phase 1 trial of this molecule, do expanded cohorts with ABA21. Now we know that we can do single agent expanded cohorts because we've already seen activity as a single agent. We're going to want to combine this with anti-PD-1 or anti-CTLA-4 in the future, with the caveat, of course, that we don't actually know, we don't have a biomarker for who will respond or not, but we'll do this empirically. And for me, the big win will be when we can combine 821 and 848 in the future, 821 and 248 in the future, because I think these combinations of cytokines, not only expanding the T cells, but giving them this effector activity will address at least some of the mechanisms of resistance that we've seen to date. And I'll show you a little bit later that the downstream plan not only is to do this combination in multiple solid tumors, but again, also combined with anti-PD-1. And down the road, for me, the big win will be when we can combine these with the T-cell engagers. Because it makes sense that if you're going to do a T-cell engager, one of the limitations I'll show you later is that there aren't enough T cells there and they're not working very well. These agents have the ability to expand those T cells substantially, not only in peripheral blood, but also in tumor, and could really enhance the activity of the T cell engagers that are currently in the clinic. So is there proof of concept that these agents these cis-targeted agents or these cytokines can work in the clinic? And the answer is yes. So there's a PD-1 targeted IL-2 from Innovent, a Chinese company, that has already shown really remarkable activity in lung cancer, in acral mucosal melanoma that's progressed off anti-PD-1. There's a really very interesting data in colorectal cancer in combination with bevacizumab. This would be the non-liver dominant group. The TGTBT stands for possibly too good to be true, because this may be too good to be true, but we'll see, and we'll see if this could be reproduced in Western countries. Recently, the Synthekine drug, which we also worked with here, which is not a cis-targeted IL-2, but it's an alpha receptor biased IL-2 made by Chris Garcia at Stanford, was studied in phase 1, had a little bit of activity in the advanced refractory disease setting. They recently did a trial in combination with pembrolizumab and chemotherapy in a PD-L1 negative group with a very bad prognosis, and the response rates were incredibly high. Not a lot of duration of response data, much higher than you would have expected in that group. I'm hopeful that will translate into real activity when they do. They've now started a randomized trial, which will be active here at Yale through the lung group. There are many other cis-targeted IL-2s, most of them against PD-1. There's all sorts of ways that you can do this by using blocking antibodies, not blocking antibodies, and really incredible, clever engineering. So in some of these cases, for example, the IL-2 molecule bends over and binds PD-1, So it's inactive in the peripheral blood. And when it gets into the tumor and it binds to PD-1, the IL-2 molecule flips off and becomes active and can bind to the cell with very high affinity. So the engineering here is remarkable and will probably really improve the therapeutic index of these drugs. So I'm not going to go through these slides in enormous detail, but I just want to explain a little bit why the combination of cytokines could be so interesting in the clinic. So these papers were published by our standards many years ago. This was probably in the late 2000s when primarily the surgery branch, but other groups started looking at combinations of cytokines, not just to grow TIL cells in vitro, but also in vivo. And when you combine these gamma chain cytokines, or you combine gamma chain cytokines with interleukin 12, you get remarkable expansion of memory and naive T cells. And you can see the graphs without showing the data in detail. This is looking at, again, at expansion. This is now with IL-15 and IL-21, same paper. You can go back and read it. But again, you can see that when you combine IL-15 and IL-21, and if you look here, at day 7, you can see remarkable expansions with, for example, you can see here remarkable expansions that you see with a combination that you don't see with either agent alone. The question here would be toxicity, but what the new technology allows us to do is to engineer these cytokines to make them less toxic. For example, this is targeting that would allow to combine these agents in the clinic. This is actually with human cells. Those were the mouse cells, human cells, showing that when you grow TIL and you add IL-12 in culture, you get better engraftment in mice, in these immunosuppressed mice. Now we can make, for example, werewolf has an IL-12 that's masked and only is activated within the tumor microenvironment. So again, will allow us to combine cytokines in the clinic that give us really remarkable expansion of these cells, improve their function with a lot less toxicity than we were able to, than we were seeing when we were giving these cytokines, wild-type cytokines in the past. So we've talked a little bit about increasing the breadth of the response and the activity of these cells, which is something we can manipulate in the clinic now. We've started those trials. The next question is, how do we improve activity in those patients who don't already have adaptive T cell responses? And there are many patients that we see in the clinic that have low tumor mutation burdens where we can't really get any activity with anti-PD-1 or anti-CTLA-4. So many years ago, we started talking to companies that had CD3 engagers already. They were active in hematologic malignancies. We wanted to see if they would be active in solid tumors. We were approached with the CD3 engager, which one end binds to CD3, the other end binds to a melanoma peptide that sits on HLA-A201. We started working with this company. We started the trial here. And I'm just showing you here that just to prove that we were involved, because they made me senior author even though I didn't deserve it. And then we sort of lost interest in this because it turned out that we had better agents for cutaneous melanoma. But the company continued to pursue this in uveal melanoma. And eventually, based on retrospective historical data, which I didn't believe at the time, they did a randomized trial of this drug against control. And you can see the survival curves here. In uveal melanoma, this is just unheard of. Very nice survival advantage. They didn't see a high response rate, although many people had reductions of ctDNA. And you see that a lot of patients that actually have some tumor regression, there was a lot of pseudoprogression. A lot of patients progressed first, and if you continue to follow them, they responded. But this agent has real activity in uveal melanoma, which is not a tumor that's very responsive to immune checkpoint inhibitors. They've made other molecules. This is a molecule that targets PRAME, also presented a PRAME peptide targeted on HLA-A201. And this molecule also has activity in the clinic. This was in their trial, the only patients where they saw activity were the ones that were actually PRAME positive, which proof of concept that they were targeting this specific antigen. So another T-cell receptor, T-cell engager, which seems to have some promise in melanoma, and they're actually now doing a randomized study in the frontline setting. And I'm just showing this slide for the lung cancer doctors in the clinic. You know, we haven't made any advances in small cell lung cancer since I was a fellow. We were giving chemotherapy as a fellow and getting miserable results. And until a few years ago, if I'm correct, we were still giving chemotherapy and getting miserable results in this group of patients. Tarlatamab is a DLL3 CD3 engager. It had a 30 to 40% response rate in relapsed refractory small cell lung cancer, and you can see the survival curves and the progression-free survival curves here. This is probably much better than you would expect with second-line chemotherapy in these patients. This led to an approval in this group. But what you see here is that despite this activity, the progression-free survival at 9 and 12 months is still not that great, 25, 30%. So there's plenty of room for improvement for these T-cell engagers. Here's another agent. This is a CD3 T-cell engager. The principal investigator of the trial here at Yale is Dr. Braun. This targets a molecule on clear cell renal carcinoma. I think it's also on some non-clear cells called ENPP3. It's a very cleverly designed molecule because this is expressed to some degree on normal tissues. They have two binding sites for ENPP3, which increases the avidity for the high-expressing tumor cells with a CD3 engager. They started the phase 1 trial. We've been involved. They started to report some activity, and this is an example. This is one of Dr. Kluger's patients who had progressed on ipilimumab, nivolumab, had progressed on a VEGF receptor inhibitor. I can't recall, David, you can correct me, I don't recall that we see too many immunotherapy responses in this group of patients. This patient really got rocked. She had a lot of toxicity with the first or second dose and probably was in the ICU, if I remember correctly. But Dr. Kluger pushed on, got the second and third dose. With each dose, the toxicity got less and less. The first scan showed mostly progression, maybe a little bit of a mixed response. These are the most recent scans. You can see really very nice reduction of this tumor lesion. It's almost gone. You can see this big mass in the retroperitoneum here that's almost gone. The response is ongoing for 10 months. This is, I think, remarkable. And we now know, I think, David, that publicly they've said something like a 25% response rate. So again, I think really an example of a dramatic result in the clinic, not the only one. There are other patients that are responding like this, but there's still plenty of room for improvement. So the question is, how do you improve the activity of T-cell engagers? Well, you can modify the construct, and that's complicated. So you have to create new drugs, and a lot of the activity could be related to balancing the affinity for the target versus CD3. Specificity of CD3 is important because as you go up on the dose of CD3, it becomes more and more specific. You get more clinical cytokine release syndrome. There are now CD3 engagers for that part of the molecule that are very specific. You can take them to very high doses, and you don't see non-specific stimulation. This is developed by the group out at in Washington. You can add a co-stimulatory molecule to this, and that might be helpful, in order to allow cells to have multiple hits against tumors. And then there's this whole issue of increasing the bystander effect. The target is important, but that's not something we can really manipulate, but it can certainly impact activity. What we can impact on is the T cell part of this, right? So this requires T cells and not necessarily antigen-specific T cells. So if we were able to increase the number of T cells at the site, we were able to increase the number of T cells, their functional capacity, we might well be able to increase the activity of these T cell engagers. This isn't rocket science. It's logical, and it's easy, something that's very easy to do in the clinic. There may be other factors that affect T cell engager activity. We can't really address those right at the moment in the clinic. It could be that you need some adaptive immunity in order to get best effects. That's something else we can address in the future. You may have to activate other immune cells in the microenvironment. Again, something we can address in the future. And maybe there are other immune suppressive factors that inhibit adaptive T cell responses. It might also inhibit these T cell responses also. But for now, we're seeing plenty of activity, or at least very interesting activity, with just the baseline construct. So is there any preclinical data that suggests that you can combine a cytokine with a T cell engager? And the answer is yes. This is data from Roche where they took a HER2 CD3 bispecific. You can see by itself, right? Some activity, not very much activity. This is giving IL-15, a cytokine by itself. This is combining the two in the mouse models. So one of our major goals in the clinic is not only to develop these cytokines, but eventually to start combining them with the T cell engagers. And we can do that in melanoma, and we can do that in renal cancer, and we can do that in small cell lung cancer. And in fact, the Asher 248 IL-2 construct is now in a phase 1 trial together with tarlatamab, sponsored by Amgen, and I'm anxious to see the safety and the activity of that combination. So one of our big wish lists here is to move forward with a combination of 821 and 248, which Dr. Kluger will somehow get accomplished over the next year to year and a half. And we've already accrued 4, and the trial just opened. We already have 1 responder, so an amazing accomplishment. And then in the future, hopefully be able to work with companies to combine these T-cell engagers, not just with tebentafusp, but with the PRAME, with the Xencor agent, and maybe even in other diseases. So still have time, about 10 minutes. The other question is are there other things that we can do, not that we might be interested in doing here that might improve anti-tumor activity? And we can't address every mechanism of action. Some years ago, these papers were published showing that in order for PD-1, PD-L1 blockade to work, you needed CD28 co-stimulation. If you block CD28 co-stimulation, these drugs just didn't work in mouse models. What does that mean? That means that that interaction is really occurring at an APC T-cell interface, not at the tumor. The tumor doesn't have any ligands for CD28, but APCs do. And I just want to point out that one of these papers was Chao-Lei Su, is probably our Chao-Lei Su, who's here at Yale now, and Ira Mellman, who was at Yale for many years and went to Genentech. So there's a major Yale contribution to this finding. These other papers were from, those guys at Harvard. Again, and what I find interesting about this is, again, these immune checkpoint inhibitors, CTLA-4, anti-PD-1, a lot of the activity may all be really in the lymph node or in the tertiary lymph node structure at that APC T-cell interaction. We're not doing enough to figure out what to do at the tumor T-cell interface. So a few years ago, Ben Izar and others started looking at these adhesion molecules on tumor cells, and it looks like loss of CD58, which binds to CD2 on T cells and is a major adhesion molecule. It's also a co-stimulatory pathway. Loss of that CD58 on tumor cells made them insensitive to T cell killing. And so there may be ways for us, and actually they published additional data. This is data from TIL, if I remember correctly, showing that in patients who progressed on immune checkpoint inhibitors, they lost CD58. So there's a little bit of data out there to suggest that loss of adhesion molecules on tumor cells would make the tumor cell more resistant to T cell killing at that tumor T cell interface. And it's not it's not a big leap to say, gosh, if you have T cells in the tumor microenvironment that aren't working very well and don't have great affinity for the peptide MHC complex, and then the tumor just downregulates class 1 a little bit and downregulates adhesion molecules, it's invisible to those T cells. They can't trigger those T cells. So there's something that we could do to make that tumor T cell interaction more effective. We might actually be able to restore the activity of some of these immune checkpoint inhibitors in a subset of patients. So some people are working on this, not necessarily for adaptive T cell immunity, but in the context of T cell engagers. This is another company which contacted me some years ago, started out of Yale Technology, although eventually they licensed in technology from Pfizer. And the scientist there, Jeremy Myers, which interestingly enough, also worked with Ivana and Andy Young at Pfizer years ago, started looking at what co-stimulatory molecules you could add to these T cell engagers in order to make them more effective. So it turns out that many T cells in the tumor microenvironment have lost CD28. They're more terminally differentiated. There's also a mechanism by which CD28 can be cleaved off the surface of T cells. So using something that stimulates CD28 might not have been very effective. But CD2 on T cells is preserved across a range of T cells, all the way from naive T cells all the way to terminally differentiated T cells. And when they did experiments in vitro and they just hit these T cells with CD3 multiple times, if you have a co-stimulatory molecule, you hit the T cells once or twice, they stop functioning. But if you add a co-stimulatory molecule, and particularly the most effective one is actually the CD58-CD2 interaction, you can continue to stimulate these T cells multiple times and continue to get cytokine production and cytotoxicity. So the thought is, if you need these T cells to be somewhat and the word doesn't come to mind, but more um, non-monogamous, I guess. I don't know. They need to hit multiple targets at the same time. The addition of co-stimulation might be, I think, an important approach. This agent, which is made by this company, just is going into clinical trials now. It's primarily for squamous cell tumors, and again, is something that we might eventually bring into the program here. And since I have a conflict with this company, also would be brought in by my colleagues. But I think it's a very interesting proof of concept, and it's another way of increasing anti-tumor activity at the tumor T-cell interface. So we've, I think, underappreciated this mechanism of resistance. I'm not 100% sure. I haven't done the experiments myself, obviously, because I don't have a lab. But it's very clear to me that tumor cells can, there's plenty of data in the literature that they can regulate class 1. We now know that they can downregulate adhesion molecules. There are a number of things that we can do to try and improve that interaction. Actually, just recently, I asked David and Jeff Ishizuka, two brilliant scientists in our group, to start thinking about this problem and to see if there are ways that we could, perhaps, for example, create a bispecific that could bind to tumor and also have a CD58 component, not necessarily with a T-cell engager, in order to try and improve that interaction between tumor cells and T cells. There's not a lot that we can do about tumor cell intrinsic resistance, but what we need is more research in this area. And I'm sure over time we'll find other ways to improve that interaction and improve the activity of T cells that are actually within the tumor microenvironment. So I'm just going to spend 1 or 2 more minutes. I just want to mention just one other approach, which is really fascinating and which we have not really started to work on here. Regeneron made a molecule that bound to tumor and also had a CD28 molecule. Now, I told you that shouldn't work because a lot of T cells in the tumor microenvironment have lost CD28. So you wouldn't think that by giving a tumor-targeted CD28 bispecific that you would get much activity. But they did preclinical studies, and they found that when you give anti-PD-1, you actually bring CD28 into the immunologic synapse. And so by giving this kind of bispecific, you're not trying to activate pre-activated or antigen-specific T cells that are already there in the tumor microenvironment. They're already memory cells. What you're trying to do is you're trying to turn the cancer cell into an antigen-presenting cell. And I wasn't sure that this was going to work, but when they took this into the clinic and got to high enough doses, this is in metastatic prostate cancer that's resistant to multiple other therapies, they saw a lot of responses. And these were durable responses, more durable than they saw with the CD3 engagers. The other interesting part about this is that this approach was associated with really bad toxicity in a small subset of patients. So things like HLH, neurologic toxicity, liver toxicity. And it turned out that everybody who got the really bad toxicity was also one of the responders. There were 2 deaths in the study, so they have to figure out how to do this more carefully. But what's probably happening here is that they're turning the cancer cell into an antigen-presenting cell by doing this. And I'm not sure that the antigens that are being presented are the standard neoantigens or cancer testis antigens. I'll skip over this for a minute because I want to just give a few slides. It turns out that more recently people have been looking into this and have found that there are these non-canonical tumor antigens. So this is basically junk DNA that's transcribed into short-lived proteins that can be presented on the surface of the tumor cell on peptide, on MHC molecules. Many of these are again, very short-lived proteins, are not likely to be cross-presented. And it turns out that many of the peptides on the surface of the cell are not actually neoantigens. They're not actually cancer testis antigens. They come from this junk DNA. So by turning a tumor cell into an antigen-presenting cell, we may be inducing new tumor-specific T cells that can not only cause tumor regression, but unfortunately, some of these might also be presented by normal cells because some of this junk DNA might also be made by normal cells and may cause toxicity. But it's a new way of thinking about this, and looking at these novel classes of antigens may lead us to new approaches, including cancer vaccines. As many of you know, I'm not a big fan of cancer vaccines, but when you're talking about a novel class of antigens that's never been presented before, it could make a difference in the clinic. So one other thing, there's this very nice work from Boston suggesting that chemotherapy, especially non-cytotoxic chemotherapy, actually is what makes cancer cells more immunogenic. Some of these cancer cells can upregulate co-stimulatory molecules. And again, maybe another way of using chemotherapy as a way of making tumors more antigenic in the clinic, and something that we could maybe explore here in the future. So I'm going to just conclude here the major part of the talk. I just wanted to sort of convey that these signaling and these engineered cytokines are really very interesting. We spend a lot of effort on these over the last few years, not just the engineered cytokines, not just cis-targeted cytokines, other engineered cytokines. We've seen evidence of activity. I think they have a lot of promise. We still need predictive biomarkers, but I still think we can see enough activity in unselected groups that these could become real therapies. I'm very interested with in the T-cell engagers. I think even the data we just saw with this one patient on the Cencor trial really convinces me that this could be a more general approach to solid tumors. And I think our approaches here to develop cytokines, look at the T-cell engagers, and eventually combine them together, it's not rocket science. It should work, and I think we can do it here more effectively. There are other things that I think we can do here, like focusing on the mechanisms of resistance, the tumor T-cell interface, maybe bring in some agents that address those mechanisms of resistance, and then perhaps work on, and David and Jeff are both working on this, novel approaches to cancer vaccines that might give us additional activity in the future. There are still lots of unexplored approaches for immune modulation. There's so many people here working on so many interesting things. I think we'll have many opportunities to bring some of these novel approaches into the clinic in the future. So I'm going to just stop there, and I'm going to just give 5 minutes of a personal perspective. I think some of you know that I'm going to be retiring at the end of the year, and I wanted to say thank you to some of the people that I've worked with over the past 21 years. I put this up there because I never felt like I was the smartest person in the group. I felt a little bit like Peter Sellers in this movie, who just nods his head a little bit and says yes, and they think you know something, but you really don't. But it's really important to be surrounded by great people, and that's the way I've always felt, that I've been surrounded by great people, and any success that I might have had is really because of the people that are around me. I want to give special thanks to a number of people. This is sort of one of the earliest pictures of our group. Dr. Kluger was 15 years old at the time. But the people that really started this group, Dr. Arian, who's now retired, but been a surgeon here for many years. He was really the leader of the group for many years, was a leader for many years when I joined. Had it not been for Dr. Kluger and all the efforts that she put in, the program would never have grown. And Dr. Halaben, who's an amazing scientist, you can see her list of publications. We've sequenced, regular melanoma, acromelanoma. She's found new pathways. These are the sort of the foundation of our program. There are other people here. I'm not going to mention everybody. Dr. Farber, who since passed away. Linda Rink was one of our research nurses who we started working with. But these three people really made the biggest difference in our program. I want to give a special thanks to Roz and Jerry Meyer. They made a transformational gift when we first started this program, and they've been supporters all the way through. But it was that initial gift that I really think got us going. And we used some of those initial funds to do, a large amount of. Ruth went around to every scientist at the institution and asked them to do big omics, and got a number of papers out of it. I'm not sure that we got a targetable pathway, but we certainly learned a lot from that initial gift. That gift was transformational in the sense that it allowed us to bring some really major scientists to the institution. Li Ping, some of Li Ping's initial package came from that first gift. As you know, Li Ping was the one who discovered PD-L1, was the first one to show the activity of PD-L1 in a mouse tumor model, and has made enormous contributions throughout his years here. And I just wanted to point out Marcus Bosenberg, we actually didn't give him the money that we promised, but but he still came. And he never held it against me that we didn't give him any money. So, and he's made enormous contributions, in the animal modeling of melanoma and other tumors. I want to point out Dr. Herbst, who came as chief of medical oncology and has been very supportive of our program. He always sends selfies. He sent me this one particularly because if you see that other arrow, that's my son. And I'm very grateful to Yale because they took him in as a surgery resident. He just graduated from the program last year. So another reason to thank the institution. As the program grew, we added a number of people. Antonella. Without Antonella, we couldn't collect all the tissue samples. Um, Laura is one of our nurses. She's since gone, but she made a big difference. Dr. Leventhal joined our group after Jennifer Choi and is a major contributor as the dermatologist of our program. Angela, who's a dermatopathologist. Dale Hahn, who unfortunately passed away, was a surgeon that we worked with for a while. We picked up Dr. Veronica Chang, who without her, we treat enormous number of patients with gamma knife radiation. Has made a major contribution. She came after Jonathan Nisley, and it's just invaluable in the clinical care of patients. And I want to mention Roy Decker, who's also passed away. But Roy was the first radiation oncologist who joined our group. Remember, people thought radiation didn't work in melanoma. We insisted that Roy join our group, and he made major contributions to our group in the early years, and we use radiation quite often in melanoma. This guy up here is not Jeremy Kormansky. That's actually Dr. Michael Hurwitz, um, who came to our group, as to see patients with renal cancer, because, we also see renal cancer in our group, and is a major part. And some of these people down here, I won't mention everybody, and I apologize for the ones that I don't mention, but Matt Burke was our nurse practitioner, and Amanda was our clinical research nurse who did so much work when we were doing the trials of anti-PD-1 and anti-CTLA-4, and without them, I'm not sure we could have done the studies here. They made enormous contributions. Destiny was our practice nurse. I can go on and on, but I don't have time to mention everybody. I just want to take a picture of our pod. I want you to know where we work. And these are, without these people, we could not do what we do. This is Adriana. She's a kind of a tough nurse practitioner who worked with us intermittently. Laura was there. Barbara Johnson, who's still our clinical research coordinator. Again, we can't do these trials without her. Amanda subsequently moved on, but made major contributions to the group. I think I took this picture because they were advising me on clothes that I should wear when I live in Miami, right? That was the picture. I guess I'm not fashionable enough for the southern climates. I want to give special thanks to Pam Driscoll, who's our administrative assistant for many years. This is another picture we took at one of the gatherings. And we managed to pick up this guy here, Jeff Ishizuka, who's a brilliant scientist working on Merkel cell perturbations of immune cells in the lab, and is going to make major contributions, physician scientists, as we move forward. I just want to say a quick word about Dr. Deepak Narayan, who's a surgeon who worked with us at the very beginning. He unfortunately also passed away, but made major contributions to our program, and I thought we couldn't leave without just mentioning him. I want to mention our surgeons, because you can't build a program without surgeons. Dr. Clune and Dr. Alino came to work with us a number of years ago. Dr. Clune unfortunately went on to Tampa, they offering more than we could offer him. But one of the best plastic surgeons we've ever worked with, Dr. Lino, an outstanding surgeon. And having that combination of a plastic surgeon and a surgical oncologist, I think made us one of the best groups in the country. I have three more slides. This wasn't a wedding. We weren't dancing the hora. This is the only picture I had where I could show Dr. Tran, who also joined our group, and sees Merkel cell and melanoma and has a laboratory program. Dr. Braun, who's one of the world's leaders in the immunobiology of renal cell carcinoma. And this is our group. And Dr. Schoenfeld, of course, who also joined our group recently and also is a physician scientist working in the lab. All these people have enormous promise, or already have lived up to that promise. What's that? I just wanted to say, so what's the point of showing this picture? Well, sometimes people would interpret this picture as saying that that it's because there's something special about the guy in the middle, but that's not really the point of this picture. The point of this picture is to show that when you surround yourself by great people, they lift you up. And that's the way I've always felt about being here. I'm surrounded by great people. I've made an effort to be surrounded by great people. And I think that just lifts me up all the time. And that's the way everybody should run their program, is just get as many great people as you can and work with them. So the last slide, I want to thank everybody that I've worked with here, nurses, fellows, colleagues, residents, administrators, administrative assistants, benefactors. A couple are here in the audience, and I can't actually say their name because I would be put in jail for HIPAA violations. The patients who've put their trust in us, and I think it's made us one of maybe the best. I think it's the best place in the world to practice medicine and to do the other things that are important to us, to conduct research, to move the field ahead. and really to teach that next generation of outstanding physicians and physician scientists. That's really what we're here to do. And this place has been amazing for the past 21 years in terms of being able to accomplish this goal. I have two complaints, one complaint. I go every morning to get my little pastry. You see it down here, which they hide behind a counter. It's like going to Tiffany and, they think that these are jewels and, you have to take them out, show them to you. And in order to get those, I have to stand behind a line of 50 people that want their macchiato or whatever coffee that takes. So I'm always late to clinic 10 days. So my advice to the hospital is please put those pastries out where I can get them without having to stand in line. And then the last thing I wanted to show is somebody, when I am going to work a little bit when I go to Miami, and I met with the chief of medicine. He says, where do you see yourself in 10 years? And I looked at him and I go, I just want to be alive in 10 years. But the other possibility is, this is where my mother was when I had to place her in an assisted living facility. I understand they have legacy admissions. So so on the way out, I made sure that they reserve a spot for me for 10 years from now. And with that, I want to thank you for your attention and happy to answer any questions.